Okay, good evening, everyone. So once again, welcome to the public talk by the NYUAD Institute. So today I will be moderating the talk from Professor Aydogan Ozcan. So this is my pleasure to introduce him. So first, before we start, I will just read a little bit about his short bio. So Professor Ozcan is the Chancellor Professor and holds the Volgenau Chair for Engineering Innovation at the UCLA. Additionally, he is also a HHMI professor affiliated with the Howard Hughes Medical Institute. He also serves as the Associate Director of the California Nanosystems Institute. Professor Ozcan has a very impressive portfolio. So with over 70 patents issued or granted in various fields, including microscopy, holography, computational imaging, and mobile diagnostics, and more than 1,000 publications in peer-reviewed journals. His contributions have garnered him numerous prestigious awards, such as the Presidential Early Career Award for Scientists and Engineers, the Dennis Gabor Award from SPIE, NIH Director's New Innovator Award, and the National Geographic Emerging Explorer Award. Professor Ozcan holds a fellowship in several prestigious organizations, to name a few: Optica, IEEE, SPIE, the National Academy of Inventors, and the American Association for the Advancement of Science, which reflect his substantial contribution to the fields of science and engineering. Additionally, he has also been acknowledged as a highly cited researcher by Web of Science, emphasizing the significant impact of his research work. So this evening, Professor Ozcan will be giving a talk entitled AI-based Advances in Biomedical Microscopy and Sensing. So let's give a warm welcome to Professor Ozcan. So the stage is all yours.
Thank you. Thanks for having me. It's my first time in Abu Dhabi, so it's a wonderful campus. I'm very happy to have joined this event, and thanks for your invitation. So before I start, how many of you are engineers or have some engineering background? How many of you have never got any training of any kind related to engineering or sciences? All right, sounds good, sounds good. So I'll try to keep it as general as possible. So today, I'll tell you some of the work that we have been doing at UCLA at the intersection of computation and imaging, microscopy, and sensing, and how these things are helping us to advance measurement science in general. The research that I do at UCLA is composed of several different pieces, one of which is mobile microscopy, mobile sensing. In a nutshell, we're trying to democratize advanced measurement tools. How many of you have a cell phone? Of course, everybody, right? So, well, how many of you have a microscope? Personal. Personal. Federal funding or any kind of funding. Personal, in your lab, in your home. Most of you don't. Well, I spent perhaps 20 years of my research at UCLA in converting smartphones into microscopes to the level where you can see cells, viruses, bacteria. For example, at the top on that slide, you see some smartphones that are surrounded with some attachments that go at the back of their camera interface to convert them into very high-end microscopy tools, with the goal that these mobile phone-enabled microscopes in your pockets could empower field workers who could not carry a bulky, expensive microscope in their, let's say, motorbikes, so that they could do field measurements with the same resolution, with the same sensitivity, specificity that you need in lab-grade instrumentation. So I was trying to diffuse the functions of lab-grade measurement tools to your pockets through smartphones. And I was lucky because when I first started, like maybe 20 years ago approximately, you know, this is your smartphone then. That was a Sony Ericsson dummy phone. That was about 0.3 megapixels, right? Do you have anything less than 5, 10 megapixels in your pockets right now? No, you don't. Well, with that evolution of smartphone cameras going to multi-megapixels, those inexpensive microscopes, like this one here, went from barely seeing blood cells into seeing viruses. For example, this one is a fluorescence microscope, dark field microscope. So it can see molecular torches that are labeling specifically bacteria, viruses, etc. That inexpensive microscope, perhaps arguably, could be a few tens of dollars to assemble, and it goes at the back of a standard camera. This is a Nokia phone. That's probably a very good camera phone that even today it's actually probably one of the best. And what you see here is how you can see nanoparticles with that microscope. In fact, you can see individual viruses with that kind of a microscope. So that if you want to, for example, look at the presence of a virus with a mobile phone, you can do. In that cell phone image, those green dots, if you close the zoom into each one of them, that's actually a virus, human virus, that is being labeled with fluorophores. These are molecular torches that light up and specifically let us know that there is a virus of interest to us there, and you can do viral load counts with these kinds of interfaces. A virus is tiny. Let me give you a context to it. Your human hair, if you just have still hair, and you pull one, that is about 100 micron, that's 0.1 millimeter. That's at the same scale of a fiber optic cable that we use for telecommunication. So that's about 100 microns. If you divide that by 100, your human hair by 100, that's about 1,000 nanometers. And a virus is about 100 nanometers, maybe 200 nanometers, depending on the type of virus. So you can see those kinds of things with a tiny attachment to, an inexpensive attachment to a smartphone. It's come a long way. When I first started, I was barely able to see a red blood cell. A red blood cell is approximately 1/10 of a human hair. So it's fairly big compared to viruses. In a matter of few years, actually, we were even able to see individual DNA molecules. This image that you see captured by this cell phone there is good for analyzing DNA. And those lines that you see there fluorescently tagged single DNA molecules, double-stranded molecules. So you could actually count them, size them, look for replication of a certain gene, copy number variations, they call it, to understand if certain bacteria, for example, has resistance, etc. You could actually use these for those kinds of measurements. In one work, we used it for targeted DNA sequencing, so reading basically mutations using mobile phones. It's been possible, so that we could look at a tissue sample with some processing, understand at the subcellular level if there's a mutation that happened at the single nucleotide level. And these kinds of systems normally require benchtop instrumentation to measure those things. So it's come a long way, empowering measurement science to diffuse into field settings, into point-of-care offices where you don't have much of an infrastructure to build those kinds of measurements that normally would take you maybe $50,000, $100,000 equipment to give you the same resolution, the same sensitivity, and specificity. So in addition to creating attachments to smartphones for the last 20 years to convert them into different kinds of microscopes, as shown here, with very inexpensive, compact interfaces, most of which are 3D printed, by the way. My lab is also very interested in utilizing the components in smartphones to build standalone devices separated from mobile phones. What you see on the left here, the battery of the laser pointer is probably dying. So let me use the pointer over here. So what you see here, you don't see it? Okay. That's okay. What it says, lens-free microscope, those are spatial types of microscopes, the main ingredient of which is the silicon chip at the back of all of our cameras. So if you open up the camera interface of your smartphones, there's a silicon chip that is the 5 megapixel, 10 megapixel, whatever it is. It's so inexpensive. It shouldn't be. It's inexpensive because of economies of scale. If you reduced, for example, the number of cell phone users magically by 100-fold or 1,000-fold, that cell phone that you have and the camera interface and all the beautiful things that you enjoy would be as expensive as a car, basically. It's just because of economies of scale. Beautiful, beautiful for us. So what we've done is we've taken those silicon chips, 5, 10 megapixel silicon chips, and created those lensless microscopes. If you look inside one of those, these are not your father's microscope, so to speak, because they don't have any lenses. So the most expensive part of a camera, whether it's this camera that's recording us or a microscope, it's actually the lens. We've been maturing lenses as scientists for centuries, right? For us to be able to see tiny things. So this is actually a computational microscope, no lens in it. That's why it's called lensless or lens-free microscopy. The main component there is what I label there as CMOS sensor. That piece of tiny stamp that you see there is at the back of all of your cell phones. That's the 5, 10 megapixel imager. Benefiting from economies of scale, thanks to all you guys having a cell phone, at least one, we're enjoying an amazing advancement in those image sensors that I can use for computational microscopy. This is as tall as an iPhone. It can generate about 1 billion useful pixels looking at specimen computationally. So this is a comparison to a traditional light microscope. Yes, it's not a full alternative to every function of a benchtop microscope, but it can do many interesting things for telepathology, for looking into specimen with very high resolution, very high throughput. Very inexpensive, thanks to, again, economies of scale. So continue buying those cell phones or other kinds of cameras. It's really helping us advance measurement science. And they're not your father's microscope. They are working based on lensless imaging of shadows. So just like on a sunny day, because of the sun, we will have our shadows on the street following us. At the microscale, any object, a cell, a virus, a bacteria, a tissue specimen, you name it, all of them have their own shadows. But those shadows are more interesting than these boring ones. They're actually textured. So these kinds of microscopes that I've shown here, they're working on microscopic shadows to be reconstructed using machine learning, using different kinds of algorithms. So basically, if you look at a tissue specimen, this is, for example, a histology slide taken from a biopsy, a breast cancer case in this case. These are the holographic shadows that you would be capturing with these inexpensive microscopes. It doesn't have a lens, so it can only capture a dummy-looking shadow. From these holographic shadows, we reconstruct them to reveal the pathology, to reveal the cells, nuclei, and all the things that pathologists are interested in seeing. So basically, from these images, it's all computational. And that's the advancement because hardware is dirt cheap. Reconstruction is almost free because you just do it once in terms of the coding and the infrastructure required. So one of the interesting applications of these kinds of microscopes is diagnostics. We looked into looking, for example, malaria-infected cells, not just for diagnostics, for understanding therapy. Are you responding? Should we change the drug, etc.? Looking into microscopic images of blood cells is still the ground truth for looking into malaria progression. On the right, you see a standard microscope. Those purple-looking things inside a red blood cell, those are the parasites that infect and explode the cell. And unfortunately, that's the parasite that is infecting us. On the left side, the lensless image is how computationally we generate the same beautiful image. Every one of those discs is about 7 micron. That's a red blood cell. That's about 1/10 of your human hair. Thus, that tiny thing is the parasite that you're looking at. So the advantage of this microscopy tool is not just it's inexpensive, it's field portable, it's lightweight, etc. Not those only. They also look at very large areas of specimen. So which means rather than looking at scanning the sample and creating large images, you can actually, in a snapshot, in a lensless image, look at many of these cells at the same time. This is a blood smear. This is one of the first methods to treat, diagnose a malaria-positive patient. Some of those cells are white blood cells surrounded by a lot of platelets and red blood cells. You need to zoom in to the red blood cells to count those parasites, and that's how we understand the parasite load and if the therapy is working or not. That's the ground truth for understanding therapy. And it's another application of these kinds of microscopes. How many of you have given a biopsy so far? So I'll tell you a lot about biopsies today. And toward the end of my talk, I'll tell you how AI is helping to deal with some of the challenges that we face with digital pathology and biopsies, biopsy handling. This is a biopsy in a thin section, breast tissue, about 3-4 microns. A micron is 1/1,000,000th of a meter, so it's tiny, very thin for a reason. And this is showing you a very large area of the tissue that we can image with these lensless microscopes. What's shown as the hologram, that's the holographic pattern that is captured with these lensless microscopes, reconstructed rapidly, giving you the same thing that a traditional light microscope gives, except there's no microscope. It's just like maybe as tall as this clicker. And it's much larger field of view. So those white rectangles, they show traditional microscopy tools in your labs, how much of a field of view area of a specimen you can look at a given time. What do? We scan the sample. It takes time. We don't need to, and that's the advantage of these kinds of tools. They're higher throughput, and we can bring in color with different kinds of techniques. This is a lung pneumonia sample, thin section, H&E stain. It's a type of stain used in digital pathology, and these are showing you lensless microscopic images where you can zoom in, continue zooming in with these lensless microscopic reconstructions to show you various different details as good as what you get from traditional light microscopy. Cost-effective, higher throughput, field portable, and great for looking into a needle in a haystack type of problem. So everything that I've shown you so far literally was coming from a CMOS imager, from an image chip that your cell phones are using today. So it's commodity, meaning that economies of scale are making these sensors very inexpensive. But you can also use other types of image sensors. These are the ones that you would find in higher-end cameras called charge-coupled devices, CCDs. For example, if you use one of those systems, you can look at a much larger area. This microscopic image that I show at the bottom there looks at an area that's about 20 centimeters square, as large as your palm, with microscopic resolution. And what you see there are some sperm cells that are being imaged with their flagellum, which is about submicron. So it's a lot of detail, a lot of throughput enabled by these kinds of microscopes. But this effort is not just trying to make microscopy great again. So it's actually also opening up some of the new capabilities that we didn't have with traditional light microscopy tools. And I'll give you some examples of new discoveries enabled with these simple lensless computational microscopes. And I'll give you the example of what we've discovered with cell locomotion, sperm locomotion. Lensless microscopy, as shown there with a simple CMOS imager coming from a cell phone, can also be used to track sperm locomotion in 3D. As the sperms are rapidly moving around, we can actually track their shadows and computationally understand their trajectories. But not just a few of these sperms, thousands of them, in a throughput that is second to none. For example, in the bottom image, I'm showing you a 10 microliter, very large volume for microscopic objects, and every one of those colorful things is a single sperm that we've tracked over something like 11 seconds with submicron localization, exactly what they're doing. This throughput, meaning capability to observe many events in 3D in a large volume, enabled us to discover new types of locomotion, precise locomotion that sperm cells conduct in 3D. Very exciting, very interesting stuff. For example, this one is a human sperm in vitro performing a helical trajectory. So this is about 1 second of its time slowed down. Color represents time, and the black thing is the sperm head. Literally, it is conducting a right-handed helix as it's moving forward, the diameter of which is a couple of microns. Very precise, a beautiful repeatability in terms of its pattern. This happens rarely. And for a free-moving sperm, this is the first time that it's observed. This was back in 2012. So it's enabled by computational microscopy. Despite its simplicity, with its throughput and precision, you can observe events like this. So this is a helical pattern of a sperm, human sperm, conducting a beautiful helix. This one is a hyperactivated sperm. It's still conducting this beautiful helical pattern, but it's irregular. The diameter is slightly larger, and it's even more rare. If you ever wonder what went wrong with your hyperactive kid, no. No, that's not the answer. But this is thought-provoking, right? So with this kind of a system, we've looked into hundreds of thousands of sperm trajectories over the course of many months, in fact, years, to discover some various interesting statistics about it. If you look at these helical trajectories, 90% of them we found were right-handed. So somehow, most of the human sperms were preferring a right-handed helical pattern, if they choose to do a helical pattern, to a left-handed one. We do not know why, but just out of Wikipedia, if you just Google it now, you will find that right-handed people versus left-handed people follow the same percentage of 90% to 10%. So of course, this has no correlation too, but it's actually a very interesting observation of asymmetric behavior of the human cells, human sperm cells, in terms of how they prefer helical sperms. We also extended this platform to look into other sperm types. So this one is a beautiful trajectory that is actually a helical ribbon. It's an amazingly organized behavior of it. This is a horse sperm in vitro being observed. Basically, the sperm cell is conducting a planar zigzag motion, but the plane is curving like a ribbon. It's a beautiful mathematical formalism that gives you this kind of a motion. We do not know under what condition why sometimes sperm cells, in this case, prefer this kind of a locomotion. But this is the precision of those computational lensless microscopes that enabled us to see these kinds of locomotion patterns. As you can see, it's a ribbon formed by these beautiful sinusoidal patterns. And it's about 4.5 seconds of this trajectory now. This one is another one. It's a twisted ribbon. Similar idea, zigzag patterns, but this one is now curving like a twisted ribbon, like a DNA molecule. And again, these kinds of observations for freely moving sperm could not be performed. Thanks to computation and lensless imaging with its throughput and precision, we could now observe these kinds of trajectories. These were some animal sperms. And next, I want to share with you something that is really exciting and interesting. This is bovine sperm in vitro, imaged by a computational microscope. The blue thing is the flagellum. So it's beating. It's very difficult to image. With holography, you can. The black thing is the sperm head. And you may be wondering, hey, what is that orange, red, and green thing? Well, that's the local coordinate system of the sperm head. Sperm's head is spinning around its own axis, like a screwdriver. And sometimes they do this kind of a locomotion. Maybe it helps fertilize an egg. I do not know. But it's a very interesting, delicate behavior, which in this case, the sperm head is moving across a helix, where at every point, it's also having its head spin. So these are, for the first time, enabled by a computational lensless microscope, which is powered by literally CMOS imagers of smartphones. This is the same manufacturers that go into your smartphone cameras. And this one is another bovine sperm. It's not now doing the twisting, it's actually sniffing. It's approaching a surface, and very much like a dog, it's sniffing to find a chemical signature maybe, or a temperature signature to find maybe the egg. So these kinds of observations were enabled by computational microscopy, helping us to shed more light on some of these nanoscale, microscale events in 3D at extreme throughput, with many of these, like thousands of these, observed in parallel. In addition to microscopy, I've been very interested in using smartphones for sensing, for diagnostics. Some examples are shown here. Early generation, not so smartphones like that Sony Ericsson at the corner. For example, that one is an E. coli sensor. E. coli is a bacteria that you can get from poorly sanitated water or lattice, whatever. It's actually a sensor that uses the smartphone camera to specifically look for colony-forming units of bacteria with very good sensitivity and specificity. So there's some molecular sample preparation, minimal, then it's using the smartphone for sensing. Some of this work has also been commercialized, so this is actually still in use, some of these devices. For example, on the bottom left, you see a cell phone with a lateral flow test inserted. That white thing right here, well, I can use my hand. So this is actually a lateral flow test. How many of you know what a lateral flow test is? You all know. You all know. How do I know? Because you all went through the COVID pandemic. Everything that was white, that you were putting your sputum on to see if you are positive or not, that is a lateral flow test. It's a paper-based test. The two main ingredients of that test were paper and gold. It had gold in it, tiny, nanoscale. That's why it's worthless. But still, there was gold in it. So early on, I created a company in 2009 to read different kinds of, much before COVID started, to read different types of tests. That device is actually a lateral flow test reader. It quantifies those signatures of different biomarkers from lateral flow tests. Lateral flow test is not, the paper-based test, is not just only yes or no, like the COVID example, which is a very simple example. It's also quantitatively telling us how much of a certain disease biomarker we have. Are you having a heart attack? Are you having some increase, for example, inflammation, etc.? So we needed readers to quantify it because our eyes are not good for quantifying those things. We can say yes or no. Is there, is there a certain disease or not? But quantification required cell phones. So cell phones really helped us democratize measurement tools related to microscopy, related to sensing, and these systems are really advanced and matching the performance of lab-grade FDA-approved devices. When I say that, I really have evidence. For example, this device over here, that one was tested with approximately 1,000 specimens from UCLA microbiology lab that were tested with an FDA-cleared device to read those, to show you that, yes, they're as good, as accurate as an FDA-approved reader for some of these tests. So this was kind of like a quick tour of some of the work that I've done on imaging, sensing, using mobile interfaces, kind of diffusing the functions of lab-grade devices to resource-limited settings, developing countries, small nursing offices, etc. And of course, AI has been revolutionizing these devices, what they can do, how they work. So everything that I've shown you had some results before the big success of neural networks, deep learning, and AI. They were already working computationally, but AI changed the game. Deep learning specifically made these devices much faster, much better in the way that they reconstruct images, form images, look at specimen. Not just in identifying objects. AI is very powerful in recognizing patterns, recognizing objects. Is this cancer or not? Where is the cancer? Where is the margin of, let's say, cancer? Those are great questions where AI can really do wonderful things. But not so obvious, maybe for some of you, is that AI can also help us improve the image. Not tell us what it is, but improve it in ways that will change the clinical workflow, make it better for the patient. How? Well, I'll tell you that story in the next few slides. So I like networks, neural networks, for this task because once you train a neural network, it's very fast to run it. That's probably one of the best things about a neural network for microscopy, for imaging, improving microscopic imaging. So for those of you who are a little bit not so comfortable with the concept of a neural network, think of a neural network as a major, big factory where there are billions of knobs, literally. The factory has a door that brings in something to be processed. It could be a potato, it could be vegetable, whatever it is. There's a door, indoor, to admit something to the factory. And there's an output. It needs a product, maybe a paste, tomato paste, let's say, canned. Well, this factory has billions of knobs that can be optimized depending on what kind of a product you want. And deep learning framework is a framework to optimize those knobs as good as it gets, depending on your quality matrix that you want at the end. This is exactly how a neural network works, except mathematically, there's ways to make sure the knobs are correctly tuned for a given task. This is a learning task, so making that factory, digital factory, correct, accurate. But once you've done it, it's fixed, and that's the beauty of it. And input goes output very fast because everything else is fixed in a neural network factory. So we've been using this idea for empowering mobile devices, for bringing advanced measurements in a very competitive way to field settings. For example, this device over here is an AI-powered cytometer. So, how many of you know what a cytometer is? Meter is a measurement unit, right? So cyto means cell, so it's a cell measurement device. So it counts cells. Every time you give a blood sample for counting red blood cells, white blood cells, 5-part differentials, 3-part differential, lymphocytes, monocytes, etc. Those samples are processed by cytometers. Now, a typical cytometer is a benchtop device that would be $50,000, depending on the throughput, maybe $100,000. But this is actually a cytometer that's the size of a shoebox. It's label-free, meaning that there's no chemistry involved in it, and it works based on AI. What you see there are plankton from Los Angeles coastal line that we've imaged with these systems for looking into content of water. So this device, for example, is taking some liquid in, as shown here, and as it's passing through this holographic system, on demand, we're reconstructing the images of these cells, these particles. So, and all of those reconstructions are using AI to reconstruct the image, not to classify them. You can also use it to classify it, but this is fundamentally helping us take a very inexpensive system and run large volumes of water through it so that you can actually identify and image objects of interest. What are some of the objects of interest? It depends on what you want to use this field portable system for. Let's say you want to look for particles in drinking water. How many particles are you drinking per liter? That's a problem for you, right? Do you have microplastics in your drinking water? We do, if you don't filter them well. So this is a great device to understand the number of microparticles, plastic in drinking water. Same with oceans. A lot of times we have algae growth in oceans that kill fish, right? If you are having a salmon farm, you must be really worried about global warming because of global warming, algae blooms might in a couple of days without you noticing might kill all of your crop. So these are tools that will enable us to label-free in an automated manner, look at the content of water and count, for example, in real time, if there is any algae bloom, what kinds of toxic algae are growing, etc. And this is completely enabled by AI because even a simple laptop like that can, in real time, reconstruct the images of the specimen as they're flowing in a very rapid manner. This system can sample half a liter of liquid, half a liter, in about 1 hour. That's a huge volume at the microscale. Every single particle there can be reconstructed. This is actually a product as well. So I commercialized this in 2016 through a company that I co-founded in LA called Lucenti. And one of the most important applications for this is really looking into quality of water and algae blooms. Another application of lensless microscopy with AI is actually looking into bioaerosols in air. Do you have mold? Do you have pollens? And how do you detect their presence specifically, sensitively, without sending samples to a lab? This is a major problem, especially if you have a flood. Let's say this building is flooded, right? What do you do after the flood? There's mold growth. Well, today's norm is this. You send a technician that will sample different parts of this building, capture some particles around to see if there's mold growth or not. They send it to a lab, culture it to understand if there's growth or not. It's a week at least during this whole period, and it's lab-based. These kinds of devices can take air. For example, there's the top part, which looks like a chimney there. It's where the air is taken in. It takes about 13 liters of air within a minute and deposits the contents of it into a transparent substrate, which is then reconstructing the images of the specimen using neural networks that are trained to reconstruct images so that we can actually look at, is there mold there or not? What kind of mold? Is there a pollen? What kind of pollen? So that you bring specificity to your measurements through AI diffusing in these field portable instruments. That one is perhaps about maybe 1 or 2 pounds in weight, and certainly field portable and very high throughput, thanks to the marriage between imaging science and AI. This is another application of the same framework for looking into virus replication. So especially when you are dealing with a new virus where you need to understand the way to treat it or suppress the viral load, drug development companies will need to do these kinds of measurements frequently. What they do is this. They take a well plate like this. Every one of those wells that look pinkish, they put cells on there. And they put the sample, which contain a certain dose of virus, to understand if the viruses replicate and kill the cells. We call this plaque-forming unit, or plaque-forming assay, or plaque assay. This can take about a week, depending on the virus of interest. For example, during the early days of COVID, many labs globally were doing these tests to understand what can suppress virality of those particles, COVID particles. And we've shown that you can use lensless imaging and AI to automate this process within an incubator. You put those wells inside an incubator, and within the incubator, you do time-lapse imaging periodically. We're looking at very small changes in the cell's appearance due to the virus to understand if the virus is replicating or not. The end result of this process is much earlier, within the incubator, without any microbiologists in the loop, we can detect the presence of the virus replication. So we can understand if your drug is suppressing it or not, if it fails to suppress the replication much earlier. For example, for this virus, we saved more than a day in incubation in an automated manner using a combination of computational microscopy and machine learning. And it works for other viruses as well. Here we saved more than 48 hours, which means compared to the ground truth that is currently used to understand the replication of viruses, we can tell you the same result about 2 days before. How is this important? Think of an emerging virus. You're trying to understand ways to suppress its infection. You need to try a lot of combinations. Time is of the essence, and that's why saving the PFU time, plaque forming time, is very, very important. And here, by looking at tiny spatial and temporal changes of cells, we can understand if the cell is being infected with a virus. Even though we don't see the virus, we see its indirect signature of cell death. So these were examples of how we have been using AI in specific deep learning to enhance different kinds of microscopes, help reconstruct images. In all of these, there's one common element, and that is the neural network, the factory that I've given you as a metaphor, as an example of a billion different knobs, right? That was optimized, the large learning model was optimized to do something that we could understand physically. It was doing some function, like tomato in, paste out type of function, and we exactly knew what it does. Hey, it's crushing the tomato, putting many tomato crushed into the same jar and then packaging it. So it was physically transparent for us. But soon we realized that perhaps we could build deep learning models or factories where input-output relationship becomes outside of this world, things that we haven't experienced before. Well, every one of us know what a paste is, right? Tomato paste is. Okay, cool. So then that's this slide. We were generating things that you can understand in terms of function, input-output. The bigger opportunity for AI and imaging science is having functions that you have never tasted before, you've never seen before. A new type of a jar, the contents of which you've never experienced. In fact, you've no idea if that jar could ever exist. That's where AI is very powerful. It creates factories that are optimized through data where the input-output relationship becomes kind of like, hey, so surprising, and I don't understand it. Good and bad. Opaque, yes, but powerful, also yes. So this is so abstract. I've lost you. I know, I've lost you, right? So, all right, come back. Stop playing with those cell phones. That function is going to save your lives. So how is that function going to be relevant to you? Well, as we age, we're going to give biopsies, and that function that I just told you, the peculiar function, is going to help you get better pathology services. I guarantee you. So every time, let me ask you this question again. This is a self-consistency check. How many of you have given a biopsy? Some people are lying, so the numbers are different. All right. So I've given several biopsies. It's not fun because when you give a biopsy, they take a piece of tissue from you, and you have to wait for the results to come back with the pathologist's report. Guess what? At least a week, if I'm lucky. That's US, unfortunately. At UCLA, a week, if you're lucky. If you have faculty colleagues from medical school, yes, a week. If not, 2 weeks. Horrible, right? Unfortunately, that's the true story. So biopsies are very important because biopsies are the way to diagnose the patient's condition. What is a biopsy? A tissue removed from our body. What goes through that process? How do they look at our biopsies? Well, a biopsy is think of it as a microscale potato of a tissue. Okay, very small, fortunately. Otherwise, it's not good. But anyways, ideally small chunk of tissue. And they cut it like they cut potato, but very thin. Fantastic. I like it. Thanks for coming. So the potato of the biopsy is cut into thin sections. Each one of those is very, very thin, like a few microns. Again, a micron is a 1/1,000,000th of a meter, tiny. Now, if I take that potato slice of the tissue, that will tell me if I have cancer or not, as simple as that. If I look at it native, by placing it under a microscope, that is what you see. The transparent stuff there is actually a piece of transparent tissue. There's no contrast to it. Because a thin tissue section, if you don't do anything to it, is like glass. You can see through it. You can see through it. Hey, guess what? For the last 2 centuries, because of this exact same reason, we are painting tissue so that we can bring contrast at the microscale, nanoscale, so that at the end of this staining process, tissue looks like what is shown on the far right. That looks colorful to you. If you put that glass slide that contains the tissue under a microscope, you can see contrast. And that contrast through a microscope eyepiece is what pathologists look and write a report on. To be able to better focus. So this is very important. This is the core of pathology for the last 2 centuries. Staining it so that transparent specimen becomes stained with contrast. Now, unfortunately, this staining process involves chemicals, toxic chemicals, that create millions of gallons of toxic waste. All of our biopsy processes globally create millions of gallons of toxic waste. Some countries in Europe refuse to produce these toxic chemicals because they are so bad. So AI, in the way that the functions that I've described, will enable us to do chemistry-free staining, which means rather than going through a chemical staining process which uses human labor, a chemistry lab with all the safety precautions, etc., and a tedious process that can take a couple of hours to a day to process our tissue samples. All of that process is going to be eliminated and replaced with AI that is learning a function that takes images of native tissue, the white, transparent, unstained tissue that looks transparent like glass to us, in another domain of microscopy, which is called fluorescence microscopy or autofluorescence microscopy, it actually looks like that grayscale image. So basically, AI is bridging the contrast of fluorescence and creating images as if it's chemically stained. So we call this virtual staining. In other words, you can think of it like this. How many of you like movies from 1920s, 30s, black and white? I like some of them, right? So AI, one of the first applications of AI is actually to color them. You take the grayscale frames of those movies and color them, looking beautifully colorful and nice, right? Well, the problem is this. I can take the same black and white movie and color it in a million different ways, all of which are different than what the actual colors were during the shooting of the film, right? I can do that, and you will have no idea of understanding. Here, I cannot do that, because I cannot create new patients. I have to be faithful. There's only one ground truth, in a sense, one patient, and I must create that image mimicking what comes out of histology. AI can do that. AI can do that. So, at the heart of this is approximating a function that we do not know if it exists. Taking a label-free microscopic image that doesn't have the standard contrast of pathologists or their training, and making a neural network take those contrast mechanisms and transforming them into equivalent images as if they're coming from a histology lab. This is actually an interesting framework that we're using, AI framework, for enabling this. This is called generative adversarial network that's conditioned structurally. Well, forget about what I said. So this is actually 2 different networks fighting with each other. It's very easy to understand, actually. Think of it, the generator, as the painter. So somebody is creating an artist painting microstructure of tissue. Those black and white images that no pathologist have any idea of how to treat the patient or diagnose the patient with those kinds of images. And that's the job of the generator. That is the artist. At the micro scale, it's painting the tissue, but in a way that is not just good looking, accurate. That same patient specimen without changing it. That's my artist. Now, artists are good and bad because they can hallucinate. They can dream of new things that do not exist. So we condition the artist to be faithful to the realities of the patient. What is the second orange-looking thing? That's another neural network that is called the discriminator. Erase that, that's actually the critique. The job of that second network is like, is this a good movie or not, not so good movie type of thing, which means the discriminator is trying to tell us if this image is actually chemically stained, coming from a best histology lab, best pathology lab, or it's created by a junky neural network. Which one? Well, this is a game theoretic approach, which means these two networks fight with each other, the critique and the painter. And at the end, painter can fool the critique 50% of the time. So which means critique loses all the power of understanding which one is virtual, which one is chemical. There's more science to it in terms of conditioning the painter to be faithful. But anyways, once I play with this factory optimization, the same factory example that I gave you with billion different parameters, yeah, I've tuned everything with this game theory fight between two networks. Then the painter is fixed. I have a painter factory then that can take dummy-looking images, like black and white. At the output of the factory, I will have virtually painted images mimicking the same process that our histology biopsies go through. And we have done this with many different samples. These are some blind testing examples, which means these are new patient biopsies never seen before. And for the first time, we're getting their specimen and painting it the way that we learned to paint virtually using AI. And on the far right, you see what comes out of histology labs. The far right images, the colorful, beautiful things, they are stained by humans, human histotechnologists that work in a pathology lab. It takes them 2 hours to stain that. Not bad. We can do it in less than a minute. More consistent. It doesn't only work for relatively simple stains. More tedious, exotic stains also work. This one is kidney tissue. New patient, never seen before. On the far right, you see a type of stain. We call it Jones-Silver stain. It's used for looking at kidney disease by pathologists. Anyone in the room from pathology? You're enjoying, hopefully? So, what does this patient have? Wonderful. Not for the patient, of course. But, so this is Jones-Silver stain. On the far right, it's actually coming from also a human histotechnologist. 3 hours. A good stain of this kind takes 3 hours. Tedious. Under a microscope, you have to inspect the chemistry. It's like a chemistry experiment for 3 hours. Well, guess what? This stain takes more tedious. It's 10 times more expensive than H&E, at least at UCLA. Per slide, you will pay 10 times more for this. Some other examples. This is Mason's trichome, another special stain, which is more expensive. The comparison is, again, between synthetically generated, AI-generated stains, rapid, consistent in less than a minute versus 3 hours later, if the histotechnologist, human, is available. Sometimes it's not, which means your biopsy might wait for the human to be available for a couple of days, and that's where AI is almost instantaneous. It also works for more exotic stains. Forget about what IHC is. IHC is very important for our colleagues, pathologists here. It's a molecularly specific stain. It looks at certain protein expressions within the tissue. And sometimes these are very important biomarkers that will be very useful for treatment. For example, what I'm sharing with you is a type of stain that literally takes 24 hours to stain. So this is more expensive, and it's the chemistry experiment with incubation and everything that takes about 24 hours. It's an overnight stain. This is more expensive. This one is very important for breast cancer. I hope you never hear this term, HER2, in your lives, but it's actually for breast cancer patients. It's a test that they have to do to understand what kind of a therapy to have. HER2 is a receptor, it's a protein at the cell membrane. This patient, for example, at the bottom has a lot of HER2 expression. That's HER2 3+. The top one is less brown. It's HER2 0. So those kinds of analysis would be done to determine what kind of therapy you should have, if it's 2+ or 3+, et cetera. So you can do those kinds of tests and stains virtually. Guess what? The failure rate of traditional immunohistochemical stains in the clinical workflow, if it's a developed country, not developed, developed country, is about 30%. In Southern California, this stain, when I go with good labs, hospitals, failed 30% of the time. It's because delicate chemistry, it fails. Human factor, human error, antibodies going wrong. No chemistry, doesn't fail. So, one sentence message. You can be better than the ground truth in AI. And that's very interesting, actually. If you filter out garbage, you can be better than what you're learning from, which means the histology lab that you're learning from makes a lot of mistakes that we do not notice as patients, but they are there. And if you filter them out in learning, you can actually be better than the ground truth. That's actually very, very powerful. 30% is not a small amount. And it's very expensive, so it's wasted tissue. I'll skip these, but hey, as an engineer, I like to show you images like image A, image B. Hey, they're identical. Find the differences between them, right, if you can. Well, that's good, but diagnosticians don't like that. They don't care. They have their own matrix. What these kinds of tables tell us is this. From the perspective of diagnosticians, we pass the Turing test, meaning that they cannot differentiate the quality of virtually stained images from state-of-the-art clinically stained, standard workflow stained images. If not, we're slightly better. The same conclusion applies from the perspective of diagnostic outcomes. If I give you virtually stained images, can you correctly diagnose the patient? Versus if I give you the standard clinical images, can you diagnose? Well, they're equal, statistically identical, which means we preserve diagnostic features within these virtually stained images. Well, it's not just matching it, it's actually transforming our ability to do digital pathology in many different ways. First, compared to humans, neural networks are more consistent. Look at, for example, this image, those 6 images on the left side. Those are human histotechnologists staining tissue at UCLA Health. You see a lot of variation there, right? That's human-to-human variation, day-to-day variation. We don't like that. Diagnosticians don't like that. AI doesn't like that. On the right, you see deep neural network-based consistent staining. This standardization is very important for giving more consistent images to diagnosticians, whether they're human or machines. And arguably, the biggest advantage of this framework is multiplexing, which means from a single biopsy, if you need a panel of stains to be performed, we can do that. Because I can take one image and go to stain A, stain B, stain C, stain D. How many of you had to give a secondary biopsy after the first one? If you ever wonder what went wrong, is destructive nature of staining. They finished a potato. They wanted to get more potatoes from you. That's the reason why we give secondary biopsies. Chemistry is destructive. If I stain it with one stain, and I need another stain, diagnosticians want to see more perspectives of the same tissue. They have to take more samples from you. We don't need to. So we don't need to call you back for a secondary biopsy because I don't deplete your tissue. And that means that tissue can be done or used for other things. So this is not just a cool exercise of an academic venture. It's actually a commercialized thing. We're commercializing this technology. How many of you know what a blockbuster means? Blockbuster is the company that went obsolete at some point. It's where you would get these DVDs, etc., to kind of enjoy a movie, etc., right? So today's pathology by and large, is like Blockbuster. We store tissue on a glass slide. If I want to observe it to diagnose the patient, I take it, put it under a microscope, very much like the DVD player, and I look at it to write my report. That's how it is today, mostly done. Netflix has revolutionized the field, right? On demand, you can get content. That's exactly the same thing. From a single image, we can generate multiple stains and provide to the diagnostician various different channels of information. This platform also works for autopsy tissue. Everything that I've shown you works for biopsy samples. It also works for autopsy. Autopsy tissue is more challenging to stain because there is delayed processing of tissue during which tissue denatures, and it doesn't stain. Chemistry fails because of delayed preservation fixation. That is a huge problem with, especially during pandemics. During the COVID pandemic, most autopsies would never stain because there were too many of them, delayed for a week. Hey, everything denatured, so staining doesn't work. Look at, for example, this slide. On the right, you have a histology, best histotechnologist human working on that autopsy sample. There's no nuclei there. There are nuclei, but there are no stains, which means it all failed because this was actually a COVID patient, delayed fixation, traditional staining completely failed. The label-free signatures, fluorescence, autofluorescence, they actually preserved even after delayed fixation, which led us to do virtual staining. So it's not just matching. In these kinds of cases, it's actually surpassing the ground truth, providing us unique capabilities. Even for delayed fixation, you can do this. So, and there are other applications of this. A recent work showed you can do staining of amyloid deposits. How many of you know amyloidosis? I hope you never hear this word mentioned to you. Basically, every part of our body, with some percentage, we store misfolded proteins in our tissue. Some of them are in our brains, some of them are in our muscle, heart. Those are bad things. And most diagnosticians would never look for them because it's not the first thing that comes into their mind with your symptoms. That's why most amyloidosis patients are treated, diagnosed a couple of years into their disease. It's difficult to find signature of tissue where there are these deposits of amyloid in the brain, maybe causing you degeneration, etc. So we've shown that we can actually do even detection of those crystals or deposits of amyloid using virtual staining. Normally, you need a polarization microscope. Most of these proteins have some birefringence. They rotate the polarization state of light. We've shown that you can actually even replace those, and you don't need to use a polarization microscope or these kinds of stains that are specific for amyloidosis. Have you used a polarization microscope as part of your service, pathology service, for looking into amyloid cases? Yeah. So those are tedious. There's no digital scanner. So you don't need to ever do it because we can virtually create those apple green birefringence, that is the signature of the protein clusters, virtually. You don't even need to ever touch a polarization microscope, which I know some of my colleagues, pathologists, hate because it's so subjective and slow and tedious. Anyways, we have also extended this to dermatology. This is an application of virtual tissue staining in vivo. This is biopsy-free. Only applies to things like skin where I can access from outside. How many of you seen that white thing touching your skin in a dermatology clinic? Do you have any uses of these devices? That's a microscope. That's a non-invasive thing. It's very gentle. It touches here and it images deep into tissue, maybe 50, 100 microns deep into tissue, as thick as a human hair. That's a device that's non-invasive, good for looking into tissue. Should I biopsy or not? Should I biopsy or not, right? And why should I unnecessarily biopsy? We've shown that those kinds of microscopes, non-invasively seeing through the skin, we can create pathology-grade, pathology-like images real-time, as shown in this movie. These are new patients, never seen before, What you're seeing here is a neural network taking those grayscale images coming from this confocal microscope. By the way, this microscope is FDA cleared for dermatopathology use, and we are showing that those signatures, as you go deeper into tissue, can be converted into pathology-grade H&E-like images, stained images. That means instead of taking the biopsy, sending it to a lab, cutting the potato and staining, you can do virtually before the biopsy, in this case in vivo, when the patient is just feeling a gentle touch on the skin. So to summarize, virtual staining is a good application of deep learning, the massive factory with billions of parameters optimized in a way that we don't fully understand what the function is. I understand the value of what it creates, but the physics of it is opaque from the perspective of imaging science. Doesn't matter, it's still very valuable. You save expert labor. Not every place would have access to good labs for tissue processing, good histotechnologies that can do staining. You can do tissue preservation because I'm not depleting your tissue. No need for a secondary biopsy. You can do multiple stains virtually on the same cross-section, a new experience that pathologists didn't have before, at the same cross-section, multiple stains. And it's standardized, which means I'm not going to confuse you. It's commercialized, as I said. Pictor in Latin, Pictor Labs is the company that I founded in 2019. This is my conflicts of interest disclosure as well, because I'm a co-founder of Pictor Labs. Pictor in Latin means painter. Yes, we're painting the microstructure of tissue. We're a painter, micropainter company. So we raised about $50 million to commercialize this technology, which is based in Santa Monica in Los Angeles. This is one of their models, Pictor Labs model. If I go back, this is a whole slide image that you see. So this is a biopsy section, a couple of centimeter by a couple of centimeter. Diagnostician would go in, quickly switch to one stain. This is the most common stain, H&E. They like to start with this one. They will go into the zoomed region of interest, scroll down, up and down, and they will switch to the next biomarker, pan-CK. It's an immunohistochemical stain. It takes 24 hours to get this. On the same cross-section, they will go somewhere else and go to another type of stain. All of these stains, if you were to do it separately, would take you a couple of days to finish, and not on the same tissue. Now you can do, in a couple of minutes, multiple stains superimposed at the nanoscale. This is Mason trichome, which normally would take 3 hours. So with this, I'd like to finish and acknowledge some of our team members at UCLA. And I want to say that there are many opportunities where deep learning and in general AI has been making and will continue to make major impact, whether it is making our measurement tools smarter, faster, better, or changing the workflow in clinical practice. I've given you an example in the especially last part of my talk from the perspective of digital pathology, how in the future image creation will fundamentally change through AI. Who's going to consume that image? Human diagnosticians, AI, and the combination. We'll see. But we don't care about that because we want to create faster, better images that are also environmentally friendly. This is deep learning based. Yes, it's consuming a lot of electricity to train and run every image. But guess what? All the negative carbon footprint is offset by eliminating toxic waste. And that's why this is a truly green AI technology. Thank you.